Okay, good afternoon, everybody. Welcome to this ICTS colloquium by Professor Anirvan Sengupta. So let me introduce him to you. He got his PhD from the Tata Institute of Fundamental Research in Mumbai in the Department of Theoretical Physics, and his PhD thesis is on string theory. And after that, he did a postdoc at the École Normale Supérieure in Paris, and after that he was at Bell Labs for a couple of years, and then he came back to India and spent a few months at the Raman Research Institute before returning to Bell Labs as a member of the technical staff for 5 years. And since 2002 he is on the faculty of Rutgers University, where he is presently a full professor. So his research interests in biology are very broad, and I have just summarized them as follows: signaling networks in biology, and biophysics, bioinformatics, and differentiated cell fates, and stuff like that. And he's the author of an upcoming book on modeling biochemical networks with P. Mehta. So Anirvan Sengupta has worked in many areas of physics, beginning with string theory. Then he changed into condensed matter physics in Paris, continued to do that at Bell Labs, and he works in communication and computation, and bioinformatics, systems biology, and evolution. So it's quite a remarkable span of things to have worked on and thought about very deeply. It's not very difficult for me to say that he is one of the two best students we produced at the Tata Institute in the past 30 years since I have been there. And it has been a privilege to have worked with him for a few years. So with these few words, I invite Anirvan to deliver his colloquium on regulating looping in genome. Anirvan.
Okay. Well, thanks a lot for such a nice introduction. Well, it was actually a lot of fun being a graduate student with Spenta. It's fine. We worked on string theory, and you'll see actually some of my work is returning. One can to that subject. One can go only so far from home, and but we also had free-flowing discussion about everything, and he kind of inspired me to take a bigger look at science beyond what you work on at that moment. So it was really privileged working in that environment when with him. Okay, so So I suspect that a certain number of people here, because there are crowd from a school, are more from physics, and I can imagine some of you are from biology. There are things where I'll try to give a very quick introduction to. Forgive me if it's sort of a little bit patronizing sounding. Okay. Well, so I'm going to talk about regulating looping in genome. Okay. What does that mean? Well, if you look at the cell and a nucleus, okay, here is the pointer. Well, this is my terrible drawing of it. Okay, the scales there are maybe 5 microns here and 10 microns for eukaryotic cells. Okay, if you take a single chromosome and you just took the linear length of just the DNA there, would be much longer, would be almost in millimeters, right? And people like to shock you by saying, oh, if I took all 23 times 2 chromosomes and lined them up, it will be about 2 meters, it will be about your height. Okay? So this is how many people like to motivate, that there is a serious packing issue here. Okay. You actually know that this is a bit cheating. It's a polymer. It's much shorter than the stretched out polymer. Polymers usually never go like that. Because of entropy, they like to be smaller. But even then, if you go through all that calculation, it's still much bigger. Okay? And in fact, if you do certain things to cells, sometimes the nucleus explodes and you see DNA spill out, right, if you change certain things. So a bit of this answer is in something that I wouldn't touch much upon, which is wrapping up DNA around histones to make it somewhat shorter. You get an order of magnitude or so there, right? Even then, it's pretty long and pretty large, okay? So, but who cares about the packing problem? Yes, in 3D volume, you can still pack it. Nothing, it doesn't violate any constraint. But the important thing is that exactly which parts of the genome talk to which parts and how that plays into regulation, there are some important things in the structure of this packing. It's not just a matter of whether you can pack it or not. And this is what this talk would be about. Okay? So, okay, so there is, eukaryotic DNA is wrapped around some proteins. Those things, usually called chromatin, they get packed further and further, and kind of here it's all artist's imagination. This picture, that if you are not into biology, this is often the picture you have of chromosomes, right? These X-like things, or maybe even half of it, right? This is the state of chromosome or for a very small part of a cell's lifetime. This is when cells are about to separate out in mitosis. The whole chromosome goes through an enormous condensation. This huge thing becomes only a few microns, okay? And you can see them as part of the nucleus. So while I was trying to steal yesterday some pictures, I didn't get very good ones. But here is some kind of electron micrograph picture. The nucleus, this is what normally the nucleus would look like. This is this whole thing called interphase, gap 1, DNA synthesis, gap 2. And then there is a short part when things are nicely paired up and separated into two nuclei. This is where you can see these black dots are condensed chromatin, whereas usually it's much more opened up, much more space filling. Okay? So this is where information stood some, I don't know, maybe 20, 30 years back, right? You knew that much. But as time goes, one is beginning to discover a lot, okay, about how really interface chromatin, genome, how is the genome organized during the period when it's much more open, okay, much more space filling and not particularly condensed, okay? So one remarkable piece of work has been to actually map where individual chromosomes are, and these are called chromosome territories. So originally when people were thinking about what happens after this little thing is got separated and two nuclei form, how do these things spread out, right? There is one picture, which is called chromosome territories, is that each chromosome still stays stuck to a little corner, right? It has its own private space, right? And the second opinion is that, oh, it will just open up and intermingle and become a giant spaghetti ball, okay? And till 1980s, there wasn't evidence one way or the other. And in fact, two brothers, the Kremer brothers, one a biologist, another a physicist, got together to test this hypothesis, and they did something very cool. They actually took a laser beam that could focus on a very small part of the nucleus, and they will do some damage there, and then they will put some radio-label nucleotides to let the DNA repair happen, right? And then they will take out the chromosome and see where have the radioactive DNA gone in, nucleotides gone in. Now, if it is a spaghetti, okay, and you did a local damage, you will end up affecting every chromosome, okay? If single chromosomes occupy a spot and other chromosomes don't come there, only a few chromosomes would get hit. So if you can focus things on subnuclear level, what was seen is that each of these damages were restricted to only a few chromosomes, giving first a hint that it's not a well-mixed spaghetti, okay? And then people got cleverer and cleverer, as they usually get. They can paint the chromosome by putting in fluorescent probes that are sequence specific, and there are all kinds of clever ways you can combine colors to read what is what. And then now you can sort of paint chromosome 1 and see where it is, chromosome 2 where it is, chromosome 3 where it is, and that kind of analysis, this is what sort of maybe the real stacked image would look like, and this is the computational solution of it telling you this is where chromosome 2 is, this is where chromosome, I don't know, wherever, right? 15 is somewhere there showing up, etc., 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 right? And basically, there are this kind of almost phase-separated structure of each chromosomes. And exactly why this happens isn't very clear. Is it because they are tethered? Is it naturally, thermally they are supposed to do? Is it because it's short enough time, long time, it will be spaghetti, but cell divides by that time? Totally unclear. Okay? So that's one kind of information about localization inside the cell that we are beginning to see in last few decades. The second piece of information that we are beginning to get is from another technique, which I'll kind of return to again and again, so I might as well give a little hand-wavy idea for that, right? So the first piece said chromosome 1 stays here. Even if it stays there, I might want to know what is the relationship between different parts of that chromosome, right? Now, there is some technique called chromosome conformation capture in molecular biology. The idea there is that if two parts of chromatin came close by, somehow get it stuck, then cut it up and sequence or something to figure out which parts of chromosome are in contact with each other, right? So if you do that, you get kind of a two-body information. The first one was what was the overall density of the pieces of a chromosome. Here, it's sort of saying what's the kind of, which part is in touch with each other at least, okay? And you will get kind of matrices like that between position I and position J. Of course, near the diagonal, you are close by, so you have stronger contact. As you go off diagonals, things fall off a little. There is a little bit of checkerboardish structure to it. Some of it is data artifact. Some of it may actually have some significance, right? So you are beginning to get a picture, okay? It is not exactly the conformation of the chromosome. You will most probably not get there in a very long time, right? And that conformation changes with time, so what's the point of getting it? Okay? But you are beginning to get statistical information about what the conformation is. Already based on that, people have seen that if I ask what is the contact probability over long distance, this is some sort of power law behavior when you go to megabases or further away. And people have tried to say, okay, if this is like polymer confined to a small region, equilibrated, what would the power law be? And if it is something that is happening in a non-equilibrium way, some part of chromatin come together, stay as a clump, then another clump contact set, etc., then there will be a much more hierarchical structure which is called fractal globule. Maybe at least the number, the exponent works out with this one. Does that say it's that? We don't know. But there is basically an effort to try to understand something statistical about the nature of the polymer conformations based on the kind of data that's coming out. Okay? Good. So this is the big picture. Now, let me go. So, I'm sorry. If you haven't known what it is, we have billions of bases, right, in 23 chromosomes. Okay? So, and a typical single chromosome is hundreds of megabases, okay, for us. Okay? A megabase is still a very small region in those chromosomes. Okay? Few megabases would have made an E. coli genome. Okay? So, now let's look at something at a shorter distance, at smaller than a megabase distance, where I would get a few genes in a region, and I want to know how are those genes regulated in this big chromosome. Now, just to contrast, I pulled out a very famous case of E. coli genome, right? This is a famous E. coli gene called a LAG-Z, and I don't need this story of this gene or this operon. But what I just want you to see is that if the gene starts somewhere here, then the regulatory sequences, the sequences that say whether this gene should be on or off, they are within hundreds of bases of the start of the gene. And people who futz around with him, they can push it a little bit, but if they put few thousand bases there, typically our regulatory site would not have a major effect. Okay? There are some exceptions in bacteria. But contrast this with an extreme case, okay? In human or mouse genome. Here is a developmentally important gene, okay? The sonic hedgehog. and it's regulated by a sequence that actually lies somewhere which is in between their other genes, right? So the sequence that binds many proteins, which tells that sequence that, a gene has to be turned on somewhere, is a megabase away. Sometimes there are other genes sitting in between, okay? So question number one, of course, is that, okay, I'll come to the questions, right? But it's already a bit shocking, right? A megabase in human beings, 0.8 megabase in mouse, and you can find many, many examples of tens of kilobases, hundreds of kilobases of distance of regulatory sequences from distance to it. Okay, so here it's a good point to at least start a discussion, and I have done something very mean, okay? Oh, sorry, what am I? I have generally made a villain and a hero and an arbiter, okay? So some of you would recognize that these three characters were the characters that, in fact, argue out the case for Galileo in the dialogues. So Simplicio, which actually is a little pun on being a simpleton, is someone who believes in Aristotle and Ptolemy, okay? Sagredo was named after one of Galileo's friends, who is kind of an independent-minded person who is willing to hear both arguments. And Salviati is effectively Galileo, making the argument for Copernican system. And when he wrote these books on dialogues, he said dialogues on tides, because that's what he thought was the great proof. And then the church told him, you cannot talk about tides. So the title got changed several times and finally became a little bit more fancy, like two systems of the world. Okay? So I'll try to use this dialogue or trialogue, okay, to set up the questions. Okay, so in my rendering, Simplicio is sort of an old-fashioned biologist who doesn't think, who basically has seen too many examples of physics failing. Okay, so they think there is always an active mechanism doing something, and most of the time he has been right before. Okay, so he says, okay, what's the big deal? These two things are inside the nucleus. They can just loop over and contact. Okay, right? So Sagredo, I imagine, as a younger biologist who is sort of more open to things. I'm being ageist here. Okay, who says, okay, suppose anything can loop and touch anything, then why not any enhancer turn back any promoters? When we knew our bacterial system or yeast systems, we could say, oh, a regulatory system close to the gene controls the gene. But now a regulatory system can control anybody, right? I mean, why 1 megabase? What's so special about 1 megabase, right? And the poor physicist says, how come? I know some polymer physics. You know, polymer theory says that, you know, if you try to return to the origin, it should most probably follow with distance as some power law. And he's sort of trying to get edgeway into the conversation. Okay, right? Okay, so, okay, the biologist sort of explains that there's already been enough debate. And these are some of the models. One of them is sort of already, he or she has already told you about, where the far-away enhancer sequence, which is protein complex, comes and touches the polymerase that will turn on the gene. There has been some people who have been worried, can it happen over large distance? They think there is a tracking model where either some protein or some signal goes along the chromatin and reaches there. And then there is a model which is linking, which is that this in-between thing gets curled up in some way. And you'll kind of see that our point of view is actually finally closer to that. But this is sort of, these are at least a few alternatives. Okay? Now, usually, once you set up those alternatives, you try to find experiments to decide between different things. Okay? And the experiments came in the following system. And I don't want to bore you with too much biology, but this is actually, I find it interesting, right? So we have hemoglobin, right? And we have different components of that. There are, so what happens is that these different components are actually not the same throughout your lifetime. In an embryo, okay, what's used often are genes that are very similar to the beta globin gene that we use as an adult. But in an early embryo, there is an epsilon globin, then there is a gamma globin, then there is a beta globin that turns on around birth and takes over, right? So we have all these genes, and they have an interesting developmental dynamics, okay, for turning on. Why is it somewhat relevant? Well, it turned out that in some patients who are suffering from some kind of anemia, okay, people ran to the gene. Let us look at the beta globin. What's wrong with it? Nothing is wrong with it, okay? And what they found out by looking at what's wrong is, again, some little piece, which later got called locus control region, about 50 kilobases away, again, across several other genes, okay? That guy has gotten messed up, okay? So it's one of the cases where a regulatory sequence far away has gotten messed up, the gene hasn't gotten messed up, and you got a disease, okay? Right? So this was a system in which a lot of things were learned about it, and people attacked it in 2002 with 2 different techniques. And in a normal talk, I will sort of explain what those are. This, unfortunately, given the time and given that this is a colloquium, I'll say take it from me that by a whole bunch of experiments, 2 different sets of experiments, people settled that something like looping happens, okay? And now, by now, in several systems, it's been seen that long-range things do physically form a loop. Now, this is an oddball bacterial system, which also does some bit of long-range affecting. And here is some electron micrograph showing occasional loop formation. Okay, this picture is sort of hard to see, but this is in flies. Okay, here is where the gene is. Here is one place that is colored in, would be indicated in blue. Okay, in a microscopic image, and this is in green. And normally you see red is close to blue and then green is far away. But when the regulation is happening, you also see sort of the wrong order. Of course, from this one picture, you cannot do anything. But if you take many pictures, you will see that red and green are often closer, okay, than it should be, okay, which signals that different parts of chromatin has come together in a loop, okay. So the evidence is gathering, okay. So at this point, we have to take this question seriously. If long loops can form easily, what stops any enhancer from affecting any gene, okay, within a big range, okay? What decides specificity now, if not distance, okay? And the evidence, the evidence on what controls specificity, right, came from a totally different set of observations, which are mostly in flies, okay? So at some point, fly guys figured out that they can have a trick to take a gene and throw it into the fly genome. Okay? They can't control very well where it goes into the fly genome. It goes somewhere randomly, right? And of course, they have put either a beta-galactosidase gene or something that turns the fly eye red, and they're hoping that it will turn on, right? And what they found is that if you take this gene and take quite a bit of neighboring region and throw it into the genome, normally, sometimes it turns on, sometimes it doesn't turn on. Okay, okay, so they are mesmerized. Okay, local neighborhood matters, chromatin structure matters, and this and that. But they found that if they bracket that region with 2 sequences, so there's a person called Paul Shadow who is in Princeton now. He found at some point that if you have genes, right, and whether the gene is on or not, you can find out by some puffing of salivary chromosomes. That if they are bracketed by these sequence elements, then you put them anywhere, they are protected from the neighbor's environment. Okay? So if they have their gene regulatory things, they are on, they don't get turned off so much, etc. So these bracketing sequences, which are called insulator because it insulates effect of other things, okay, has some magical property of protecting you from effect of the genome. Now you should already wonder, okay, why would it do such a thing? Because the polymer can loop over and touch over anything. So having a little sequence in between should not do anything, right? Anyway, as time went, people found more insulators in flies, and in vertebrates there is actually, so far as we know, there's only one kind, but insulating sequences are there, right? If you look at gene expression in flies, quite often you see clean boundaries of correlated expression. Okay, and those boundaries often correlate with presence of insulators at the boundary. Okay, right. So, well, I gave you the models of long distance action. So people were prompt to sort of figure out corresponding insulator models. Well, how would insulators work? Well, one idea is that insulators are like fake promoters. That steals that enhancer and doesn't let the real thing there. Or it's like a blockade if it is traveling along this that whole path, right? Or just like, again, this is like a fake promoter, right? If this is a mechanism, then having two insulators there would be better than having one insulator. Two doors are better than one door at blocking, right? Two stealers are better than one thief, right, at stealing. Okay? Remarkably, what ends up happening is that if you put two insulators next to each other, the regulatory sequence can talk to the gene again. So, voila, okay? All this thing sort of going down the drain, okay? So, so there's a whole bunch of experiments which seem to suggest that what's happening is the following. Usually, if between enhancer and promoter there is one insulator, that insulator finds a friend somewhere else, right? That actually ends up bracketing this enhancer and doesn't let it talk to the promoter. Okay? So there is some strange bracketing game here. If they are together, these two come together and they're like a little loop on top of chromatin, right? So it separates out this region from that region, but it doesn't quite have any effect. Okay? On the other hand, if you put two sequences bracketing the enhancer by hand, right, then it stops its effect. Okay? So this is where we are, right? There is a hypothesis developing without any explanation how physically this would ever happen, which says insulators like to pair, form independent loops. Independent loops don't talk to each other. Okay? Now the question is that how can ever such a thing be true? Okay? Okay, so our three characters return again. Okay? So, well, it's clear that, if I make this loop domain idea that pinching creates loop domains and loop domains stay independent of each other, then that's a neat hypothesis that explains a lot of data. But, this guy correctly points out that it's very non-intuitive that if I have two loops like that and they are fluctuating around, that they would not be in contact with each other. Why not? Okay? Right? And somehow, so here is the biological physicist who's saying that I have two problems, right? One is, why is it that I have efficient long-distance contact? And the second problem, which is that even if I have efficient contact, if I make these topological domains, that they don't interact with each other, so maybe two problems are better than one problem. Sometimes one cancels off the other. So if I can understand one, maybe I'll understand other. Okay? Because there is something more mysterious than simple flopping loops. Okay? Okay. So this is where perhaps we enter. Okay? Normally, if you take a polymer and ask it, what's the probability of it turning and looping back? Well, there is some kind of stiffness dominated region in short distance, and then it kind of falls off. In some regime, it looks like Gaussian walk returning, which is 1 over separation to the 3/2. And at some point further down, maybe there would be some effect of interaction, polymer interaction. DNA, for example, would behave very much this way. Okay? So we're sort of thinking now that it's not just, maybe chromatin having some degree of interaction with itself helps. Okay? So we pretty much blindly started working on assuming that chromatin has tails that could actually interact with DNA, providing some attractiveness. Okay? So, these are our little theorist chromatin with little balls with occasional bond stitching. And you play with it and you don't know what you are doing, really. Okay? But and most people who have seen other work with polymers, okay, would have to realize that we are working at a megabase scale, which is much shorter than much of the other studies that have been done, okay, over polymer looping. Anyway, turns out that there is a certain parameter regime where attraction does produce a slight enhancement in the long distance of contact probability. So you can kind of see it there. This would be without any attraction. This would be with attraction, right? And it is about factor of an order of magnitude higher, although both are small. Okay? And an order of magnitude higher is good enough to explain the gene expression enhancement we see. Okay? Right? But that's okay. Now you could say, well, what's the big deal? You made a polymer, you made it attractive, well, it will collapse sometimes, right? And that will enhance probabilities. Well, so I'll claim that it's actually a little bit more interesting than that. Okay? And later I'll tell you it's even more interesting. Okay? So the first thing is that you also have to know, if I go back a step here, that people have quite tried to do measurements of how genomic distance map into physical distance. So they put a little painting spot here, another painting spot there, measure distance under microscope, and you know the genomic separation. And you know, if you wanted to fit it with a square root something, you would get away with it. Okay? Which kind of says that it's not quite like a collapsed polymer. Right? So random walk for short distances is not such a bad description. So how can it be that I'm getting enhanced contact, and yet, okay, it is random walk like if I take two generic points and look at their separation. Okay? So the answer to that is looking in greater detail at our simulation. And what you see is that in the parameter regime we are working, okay, most of the time there are only a few contacts in this polymer, right? On the other hand, occasionally you get these very large clumps, right? So if I have a finite size polymer, it doesn't always have to choose absolutely between a collapsed state and an open state, okay? This one is actually going in and out of it. So that's very, very good, because that way I can have it both ways, okay? The typical separation between 2 points would be given by the open configurations. That's what the dominating contribution is. But return probability in that case would be dominated by these configurations where things are much higher density. Okay? Fine. So if the story ended there, right, then okay, what we have done is to give attraction, things collapsed occasionally, that brought things together and enhanced the probability. Big deal. Okay? Right? But something more interesting happens in this, where you try to understand how insulators work. Okay? So now you do the numerical experiment where, so this is just all statements that indeed it's the collapse configurations which is giving the enhanced interactions at large distances. But it is not a permanently collapsing, it opens up again, right? So it dynamically goes back and forth between these structures. But what we want to really do is something more tricky. I want to take a loop, I want to break it up into 2 loops. Of course, the computational version of this is these 2 loops, right? And I want to see a point, if I go along the polymer on the other topological domain of the loop, how much contact I get, and if I go same distance within the domain, how much contact I get. Okay? So I want to now compare, okay, inside topological domain contact and for same distance outside topological domain contact, and see whether there is a difference between the two. Okay? And what you see is that actually there is a difference. Okay? So for some reason, okay, this is not what it is like. There are much more contacts inside a domain. This thing is a permanent bond. That means pinch these two and keep it pinched. This is a contact map. Okay? You rarely get a contact that is crossing this one. Okay? So, okay, happy. I could sort of say, well, we are seeing insulation effect as well, right? But I am a little bit uncomfortable because one trouble with simulation is that you are doomed to succeed. Okay? You work and work and work and work. One day something works, you declare victory, right? So I'm very nervous till I have some other ways of understanding this. So whatever little time I have, I'll try to say something about what we understand, and perhaps it can form a new hypothesis that could possibly be attacked experimentally or checked experimentally. Okay, so here is the idea. Okay, suppose I take my simulation and I draw a contact map, right? I want to know, are these contacts such that there is a lot of crossing or there is no crossing? Okay, why am I going there? Well, if there is no crossing, say contacts are always this way, and I make one crossing permanent, right? Then that says that these two sides don't interact with each other, right? Because I never cross a bond. Suppose, I mean, of course, statistically, there would be some crossing. But suppose I say there's an absolute rule, no contact cross each other, okay? Then I actually have an explanation why pinching, okay, makes this domain separate. And I'll make it a little bit more plausible in a short while. So first observation we have is that if I sprinkle random bonds of the same length distribution and see how many crossings I get and see the number of crossings we really get, you see that this is lower, okay, right? Significantly lower. Fine. What does that mean? Well, imagine there is no crossing. Then what kind of folded structure would I get? Well, I would get folded structure that look very much like RNA, right? There would be stems, stem loops, right? And if I open it up, this string up, and I draw the contacts, and this is my terrible effort of drawing it in short time, they will be like these rainbows inside rainbows, okay? So here I come back closer home, right, to my PhD thesis on planar graphs and non-crossing diagrams. Okay, so yes, so if this is true, and I say, well, I'm going to make a permanent bond somewhere by making one of these bonds stay always, then there will be loops inside and loops outside, right? And the domains should be separated automatically, okay? Fine. So, so this is getting somewhat more interesting. So you might ask, why is chromatin not having too many pseudoknots, just to use an RNA-like language? I have stem loops, but stem loops don't bend around and kiss each other. That's called a pseudoknot there, okay? Right? So the explanation why it is somewhat suppressed is that if I do play the following game, I give you some polymer. I ask you to put n contacts there, okay? So I have a fixed number of interactions, okay? If I put n contacts and I ask you, I put it randomly or I put it like stem loop, which one has higher entropy? Typically, the stem loop one has more entropy because the polymer, each of these parts can actually move independently, okay? The crisscross one turns out to have lower entropy. Now, why is it not that collapsed polymer is thermodynamically long polymers collapsing isn't like stem loops? That's because at very long distances, there are many more ways of doing crossing diagrams than non-crossing diagrams. So finally, each crossing configuration have lower entropy, but there are many, many more ways of doing crossing structures, right? So planar diagrams, there are only so many of them, okay? So as a result, in thermodynamic lengths, this doesn't happen. But we can still have an intermediate length scale where we see stem loop-like behavior rather than this one, rather than the collapsed polymer, okay? So again, you can ask the question, okay, answer this original question, why are two flopping loops not interacting, okay? So the answer to that is a little tricky. The answer is that actually two flopping loops do interact. But remember, in the open configuration, long distance contact is very unlikely, right? That is like 1 over distance to the 3/2. And so these loops touch, but over a megabase, you get very little contact. The contact is dominated by the collapse configurations. And if the collapse happens like stem loops, then separated domains don't intermingle, okay? So in some sense, if polymer contact is dominated by these ones, it's really suppression of this and having more of these configurations that has caused our results, okay? So anyway, so I want to say, you could ask, what is the prediction from there, right? It's all very good to have a theory. Do you have a prediction, okay? And the fact is, we do have some prediction, even if this is a model without any parameters, very general, very, very kind of qualitative picture type. So one thing we are saying is that when 2 points come to each other, okay, quite often it happens because the whole region is much smaller. Now, if you have multiple fish probes on the system that can glow, right? And you can do 6, 7 colors easily these days. If 2 of them are close to each other, you have higher probability, okay, of seeing many other things are close to each other. So question is that if I do some high throughput imaging, okay, of cells with multiple probes, do I see in some sense higher abundance of multiple things clustering together. Okay, that's one. But there is an even more interesting one, and this is an experimental method that's coming up, most probably getting published soon. Remember I talked about this chromatin capture method, which says 2 parts of chromosomes together, right? Usually that method is averaged over many cells. And inverting that to get the conformation is a non-trivial problem. We actually work on that, and maybe I'll briefly mention that, right? But people have tried to take the method to the next level, where they take a single cell. They do crossing, right? They try to cut it up. They do some grand horrid PCR or something, right? Not PCR, actually sort of whole genome amplification out of there. And then they try to figure out which parts are with which are, right? So if you get the good mapping there, right, you are getting something about contacts in a single cell, right? And the contact matrix, which I'm sort of ordering always I less than j, so only 1/2 of the matrix, right? If you have a spot there and you are non-crossing, then that constrains the spots, other spots to be somewhere, right? So sort of these two would be disallowed. They will correspond to this kind of thing. These would be allowed, okay? So it lays down a constraint on what kind of configurations I will see in a single cell, okay? Chromatin conformation capture data, okay? So statistically, how to analyze this is actually a very fascinating question, and this is something we want to do. Okay, so I want to briefly say that we have come out there and tried to understand. Take a step back from the simulation. Okay, let's try to say we reformulate the original hypothesis. The original hypothesis, as formulated by biologists, were that if I have insulators, things actually clamp, and somehow topologically independent domains do not communicate very well, right? I want to reformulate it and say that the topologically insulated domains do collapse in this stem loop way, which makes polymers look like branch polymers. Okay, and there are occasional violations of them, but that is suppressed. Okay, pseudoknots, just like in RNA, you would have said pseudoknots do happen, but not as often. Okay, so does it happen because it is, equilibrium dynamics over some length already produces that? Okay, it's a possibility. It's possible that you really have to think of short time dynamics of polymer in a mesh or spaghetti of other things, and you can imagine that you can go through, come back, go through, come back again. But actually, there is some hope that we can understand it differently. Why? Because if you look at attractive polymers, superficially to first order, it looks absolutely like quantum mechanics of a particle in a disordered medium. Okay? So if you write down the field theory of each, it will look very same. Now, why is it that they are not exactly the same? Well, you have to be careful. Attractive polymers need a second virial coefficient that's negative, and third and fourth and other virial coefficient that's positive, okay, to stop it from collapsing to nothing. If you take a disordered system where the potential fluctuation is only Gaussian, of course you get a density tail that goes to -infinity, right? The way to make the two things the same is to have a non-Gaussian potential distribution that cuts off the low energy part, okay? And in those, they are exactly equivalent. The field theory is exactly equivalent. Now, any of you who have played with quantum disordered problem know one way to do theory of metals is to do a large N expansion, okay? Organize planar diagrams first. Those are how the Green's functions look. That you start doing ladders with it, you get your conductivity, right? And large N expansion is a way to organize weak localization corrections, okay? So you kind of do the same thing, right? And what you see is that you can now think in terms of what I will call the genus of a contact map. Okay, you look at the contact map, stretch it out like that, make it into strips, and there is a way of actually thinking about it, whether they are planar or non-planar, or how many handles of surface you need, right? So I can organize my perturbation theory of the polymer that way. We're trying to understand how that works. But also, suppose you forget all this field theory, right? You come back and say, can I use it for data analysis? Yeah, give me a single molecule contact conformation thing, or give me a simulation. I can now start analyzing what kind of topology do you get of contacts. Are they crossing, are they non-crossing, right? And you can do something more than that. So there are things to be pulled out of data that is kind of doing statistical topology, right? Which is actually a growing way of analyzing data, where you do not want to understand, okay, exactly whether this was Gaussian or chi-square or something or the other. You actually take different length scales, you get a mesh of things which are connected to each other, and you can actually calculate things like homology groups on it, right? And as you change the size, you do renormalization group, your homology group generators change, and that actually tells you about persistent topologically non-trivial structures in a system. Okay? So this has been actually a method that's been used for data exploration, both for structural biology and other things. Okay? And you can actually very relevantly use it in this context. Okay, so I started about 10-15 minutes late. I'll most probably not take this too much time for you, but I would sort of say I could also leave this idea of trying to understand what polymers or chromatin as a polymer do in general, and also look at particular things by taking polymer physics a little bit seriously to inspire probability models of the data. Okay? So how do I do that? So there are tons of data of this kind these days. This is some Hox gene locus. You have little fragments of DNA there, and some experiment tells you this big black dots, meaning the corresponding 2 points are in contact with each other, right? So you have some observation, some number which is absolutely free of any scale or anything in arbitrary units. You have gotten high contact, low contact, high contact, low contact, okay, throughout a regime. You'll get a matrix of such numbers. Okay, so one of the things that you do is to look here and say, this is a high contact, maybe this is significant. Okay, let's draw a contact. Okay, that's one thing, detecting contacts. So there's some kind of significance test maybe. The other thing is that, okay, if you tell me all the contacts, right, what kind of conformation is that, right? I might ask, just because you got a high value, does it mean it's always attached? Okay, or is it attached 50% of the time? I have no way of putting a probability back on that, right? It is something that is absolutely arbitrary number at this moment. Okay, so people have still tried to do something, and one of the things that has been tried is some people said, okay, suppose between I and J there is some contact strength, Cij, okay? Let's try to find a model where that relates to a conformation, 1 over distance to the some power fits Cij the best possible way. So you try various polymer conformations, you say, okay, this is the one that comes closest to satisfying Cij is proportional to 1 over distance to the alpha, okay? And this is practically stolen from how people do NMR structures, okay? You can see interactions in NMR by looking at the shifts, right? And you can make an estimate of distance, and a lot of protein structures were done by saying this is close to that, that's close to that, how could you get a structure, right? The problem is that chromatin, especially this is yeast chromatin, it's rather dynamic, right? Nobody says chromatin actually, these chromosome 3 shapes has the shape of this Arabic letter, and then it kind of freezes there forever, okay? In fact, you can tag things, and this is not a very good way of saying it. You can actually see them move in live cell imaging, right? You can see 2 points come together, fall off, come together, do all kinds of things. Okay, so we would rather try to go away from it and take a probabilistic model, okay, of polymer conformation and try to solve this problem. If I see a bump in that matrix, is it a nearly permanent contact or is it 10% contact? How do I do it? Okay, so here is the intuition about how you do it. In this famous matrix, right, there are things that are near the diagonal, okay, and there are things that are off diagonal. So off diagonal things are what? Well, you see if 2 parts were joined, of course those 2 would give a big signal in 3C because they are actually close to each other, say 50% of time, right? Then somebody who are neighboring those sides, they will also give a bigger signal than usual because 50% of the time this is really pinched, okay? So if A and B are pinched, then C and D actually have a shortened distance in between, right? So I will see as if, okay, this is a polymer correlation fall-off structure, right? Except that this should be typically weaker than what's happening on the diagonal. Because in diagonal, 2 points of a certain distance, same distance, and C and D are always tied, okay? So if I take neighboring regions on the chromatin, they should give a strong signal. Very far away of the region, If they're giving a strong signal pattern, that's because some percent of time something was pinched, and close by areas are getting you same pattern, right? So actually, if I have a principled way of comparing what's the pattern is near the diagonal and then try to fit this part, I should be able to pull out what the probability is, right? So if you do it in a slightly fancy way, but it's not very fancy, okay, you can sort of use a kind of maximum entropy kind of approach to say, I know these moments of contact, what kind of probability distributions can I get? And okay, lo and behold, you will get up interacting heteropolymer model. Okay, that's just window dressing. Okay, but when you try to fit that model, you start, okay, so we do some tricks. We don't want to do polymer simulation. We actually use Green's functions to estimate these things. Okay, but this is the kind of thing you can do. You can, right, start with data, see whether a pure free polymer model fits it. If it, wherever it doesn't fit, I try to introduce interactions, and then I keep on going. And here is sort of chromosome 3, where we, at the end, we are sort of trying to say what is the percent of time these pairs or that pair are together, and what are also the polymer pinching configurations that you get out of here. So in some ways, you can use that to actually process data and say something about whether some interactions are permanent or transient. Okay? And we actually get some somewhat shocking things, which I first tried to tell my student that he must have done some mistake. Okay? And it's something to do with East chromosome biology, where there is a mating locus here, which is supposed to get some genetic information from this hidden mating locus. And we found the loop to land somewhere else. And I said, oh, you got something wrong. It should land exactly there. And then we found that actually there is a sequence element little bit away from the real thing, which is crucial, which is known in the literature to be crucial for a mating locus and hidden mating locus stocking. So actually this 11% bond that forms here is in fact in the right place, right? So there is some shock there. Anyway, so I want to sort of summarize what we are learning out of there. So if what I want to say that there is a potential hypothesis that chromatin does fold in this environment, okay, from time to time. But when you get the folded conformations, the contacts have suppressed number of pseudoknots. If you want to organize chromatin like planar conformation, 1 pseudoknot, 2 pseudoknots, 3 pseudoknots, right? So the amount of crossing is lower than you expected, okay? And that is a hypothesis that you can try to test. You can argue with me whether equilibrium polymer dynamics produces it or not, okay? Or we need something more fancy, okay? I'm open to that, right? But if you set that up as a hypothesis, you can possibly explore it, okay? So that's one, okay? Second is that thinking of it this way, it's a very powerful way of looking at topology of contacts, because we are not often being given the exact polymer conformation, except in simulations, right? But if you are going to get single cell contact data, we can take that, we can take different length scales, and when we take different genomic distances and glob things, we can see how contacts disappear, right? So it tells us, for example, whether the pseudoknots are short distance pseudoknots or long distance pseudoknots. It gives us all kinds of statistical ways of analyzing this. And basically, there are lots of data coming out. I talked about fish data. When I talked about fish, which is fluorescent in situ hybridization things that glow that can be hybridized with particular kind of sequence of DNA, right? And I talked about it in the context of painting whole chromosomes. But you can actually do local painting and see how distances are fluctuating, right? And so far, very little has been done about which framework within which to analyze this data. And here is a good opportunity where you can set up a very basic polymer model and make sense of many things. Okay? So, okay, I've done pretty well on time. So, I should thank my collaborators. Some of this work was originally done with Swagatam Mukhopadhyay, who is right now working in as a computational biologist in Cold Spring Harbor. And these are a series of students who have also worked on this problem. I learned a lot about this problem from Vasily Studitsky and Paul Shedel, and there are several other groups in Rutgers who are also interested in this question, and I have benefited from interacting with them. So, thank you. Yeah. Mm-hmm. Yeah.
Can you connect it with the regulation of the expression? Because in a condensed chromosome,
Mm-hmm.
if I have to express it at opening,
Yes.
so I can have a localized opening if the contacts are in.
Right. So it is true. So actually, fortunately now there are even few cases where you can directly image the process you're mentioning. So I hadn't quite said that, but if you go to fly genome or something, you do see that there is a repressive state, a chromosome which is sort of even in interface, it's not that open. And then when you open up, the whole part kind of becomes more fluffy, and you see something. So I could have understood the explanation that perhaps it's small enough, but we can almost measure how big it is and how much contact there would be, okay, in a naive model. But when it opens up even, right, maybe just because it's close by that those contacts happen, that the original condensation itself is the reason. But that still doesn't explain why insulators work, right? Because I can open up this whole region and you have an insulator, and it's usually opening structure isn't different. You can have different enhancers talk to different promoters in that neighborhood. So I think there is something more than just opening up.
And no contacts will cross each other.
No, this is crossing. Of course, every contact in 3D, you can call it a crossing. It's just that, so this is a slightly tricky concept. And the idea is the following, okay? Imagine I have some 3D polymer, right? And there are some, okay, I should draw things with more contacts, okay? What I want to do, okay, I don't have another color, okay? You imagine you attach rubber strings here, wherever there is a contact, okay? Maybe another there, right? And then you stretch open this polymer, okay? Right? And then what you try to do is to have this guy go up in the sky and fall down, okay? On the same side of things, okay? So I most probably already have gotten one crossing here, and yeah, the other two. So I'm going to, so I wish I had two color chalks, but the idea is that there is the main polymer, and then there are these rubber bands sort of going out and coming back to it, okay? And I want to know what's the topology of crossings here, right? And that can be done. So actually, if you are a little bit computer sciencey, you must have heard that the problem of genus of a graph is actually an NP-hard problem, right? But this is actually a much easier problem, because this is not the lowest genus surface in which the graph can be put. This is what, actually this must have a name, but this is basically the strip genus, in the sense that I fatten this, I fatten that, and I go, right? So there is a certain relationship between, okay, so imagine that strip passed under or over, doesn't matter, okay? If you want to draw it on a surface, there has to be this orientability put into it. Then suddenly there is a genus associated with it, okay? Maybe for better job, I should actually end up making a circle out of it. And there's a well-defined object you can define there. And I don't think anybody has carefully looked at it, okay? In RNA context, Tony Z and others have written a bunch of papers connecting large N expansion and this, but you can actually now use it for real data.
Contact map is happening spontaneously.
Yes.
And then you sprinkle your insulators.
Right.
So, but even then, the contact map, this is a statistical statement. So
right.
That still doesn't show why the insulators work reliably.
Well, insulators, actually there are several experiments. So first of all, insulators absolutely don't kill. It's not that there's no contact at all. You might end up finding the gene expression has fallen off a lot, okay? And there are some claims, if you believe what I'm saying, you don't have to, right? You would actually say, if near an insulator things are very close by, that may not be stopped so much by all this cost. Okay, and there are some evidence where if things are not really long separations, that it is not reduced. Okay, so again, the effect of the insulator doesn't have to be perfect. It has to most probably bring some. So if you saw these pictures, here is a fly gene called white, which is actually the one, as you know, makes the red pigment for the eye, right? And you do see lower expression, okay, even with insulators. Okay, sorry, I'm going to be embarrassed here. But somewhere down there were poor flies lying down, okay, right? Okay, so if you got, for example, from something very red to something whitish, but yellowish, orangish, okay, that's good enough. And even in the simulations, it's not it's zero, okay? It's just much less than what would have expected. So, okay, so let me go back and explain something else that was the bigger picture before, which is happening for the whole chromosome, okay? If you squish this chromosome in a restricted volume, right, and let it be for a long time, what would happen is that the end of the polymer would sort of entangle itself and make this kind of condensed object. Or you give attraction and you get a collapse kind of globule state, right? In the globule state, you actually have much greater topological entanglement, okay? On the other hand, if you let a polymer collapse under short time, quite often you get a hierarchical structure. You get a blob here, and actually the hierarchical structure most probably has less crossing in the same sense than the globule state, okay? So there is something to be understood, right, about polymers under different conditions and chromosomes and their contact structure topology. And I most probably would say that's the message out of here, that certain efforts are producing something like that in a simulation is encouraging, but you shouldn't take it as the final word. Okay.
If I understood correctly, what you said is that if I'm putting insulators,
Yeah.
into small loops,
Yeah.
and those small loops will interact with each other, I mean, within themselves rather than acting.
In the loop, there will be interaction. Things outside the loop can interact with each other, but things inside the loop doesn't seem to interact with things outside.
And the first part is that there is a mega phase distant thing that has to come and meet it.
Right.
So that was like the simulation shows that
There are some collapses.
probability is,
Right.
I mean, it's still high even at long distances.
Right.
But contact probability is higher with medium distances. So even here we can form two loops, right, which should be more likely rather than one. And I don't understand why your mega base will still come and get in touch.
Oh, the mega base, the reason it will come in with some higher probability and touch is that occasionally these things have started making these stem loop structures. And when you make the stem loop structures, okay, so if you just had stem loops, okay, then the system actually looks like a branch polymer, okay, and the size of the system doesn't go as length to the half cubed, right? That's the volume in which usually the tail is, right? If I had just Gaussian walk, okay. In a branch polymer, that length scale comes down to n to the power 1/4, okay. So it's a much smaller volume, and the probability of two things being in contact with each other is much higher.
But still,
Okay.
I mean, the middle thing is more probable to get in touch with. So it's like these two ends are there.
Oh, so you are saying that there should be some distance dependence even in this model. It's not going to be completely flat.
It seems that if there are these 2 points far away which have to come close, there
Right.
can be a point in between where two small loops will form, like the way it's formed in insulators.
Okay. Yeah.
Insulator kind of structure is more likely to form even without insulator.
Oh, yeah. Very good. Good. I understand your question now. Okay. Yes, it will form. And then they will not, okay. Actually, the two ends can still interact with each other, right? Okay. Because middle insulator doesn't stop it. But the more important thing is that those are not permanent contacts. They form and melt. Okay. So there will be things that will look like insulators, even inside the freely associating thing, but these bonds are not permanent. So they come off, and you get a rearrangement. So sometimes some things would be in contact with each other. So, okay, if I could restate your problem, this is what most probably you are saying. Okay. Suppose I had this contact form anyway, okay, just a bond. Then suddenly this guy cannot talk to this guy, right? But this is only as long as this bond is there, if you take this rule seriously, okay? But this bond would open up at some point. So allows, but if I put an insulator there, really, that's a permanent bond, okay? So it's the permanence of the contact that makes the difference, okay? In the simulation, we would form those bonds, and they will separate some domains for a time being. Okay? Because if I have this kind of stem loop formation, then usually this guy would like to form like this, and this guy would like to melt like that, right? And they wouldn't usually, they have to have branch collisions for things to, right, come together, which has lower probability. Okay? But, okay, so maybe we'll do that offline after that, okay? Right.
So typically, I guess they are sort of equilibrium calculations.
These are equilibrium calculations, yes, and that's actually one of the headaches. Yes.
It's a kind of highly non-equilibrium situation.
Okay, so what my take on this is that I'm totally with you that the whole chromosome is very unlikely to be equilibrated in such a small space, right? Even if we have separated globules, it most probably hasn't gotten. Question is that whether a shorter segment has gotten time to equilibrate. So you can do all kinds of either Gaussian or repression-like or whatever estimates, right? Depends on how you are assuming the microenvironment to be, which we don't know very well, right? But as you know, that the answer for relaxation depends very much on are there other things going through? Is it in a melt-like situation or not, right? So we really don't know what's the local environment like, so that a megabase, can we treat it with equilibrium? And the other thing is that suppose it's in the other regime, that it is very entangled, right? Then there may be a totally different reason for being like branch polymers, right? Because, for example, in reptation model, what happens? You have a tube and you stay inside, right? Now, suppose you want to deviate from there in short time, okay? One thing you can do is to just snake through. But suppose you have like a herniated situation, something tries to go on the side. Usually two things will go together. And you will in fact get a branch polymer. Okay, we can discuss this. It is again related to this question that for polymer physics, superficially there is a great deal of similarity between polymer in a disordered medium, okay, which is very similar to quantum mechanics of a particle in a disordered medium, okay, and right, polymers that's interacting with itself. And there must be some subtle differences, but superficially, a lot of arguments are similar in this case. And in some non-equilibrium cases also, you can make the stem loop-like contacts more easily, okay? And I can argue sort of how, right? So I guess question one that I don't know is that it is a short enough piece, okay? Can that be like equilibrium dynamics, okay? Right, effectively. And the other question is that suppose we don't have equilibrium there, even then can we get stem loop-like structures, okay, for non-equilibrium reasons itself, right? And I think both are worthwhile things to sort of analyze, simulate, do some theoretical calculations, and maybe even observations. Okay? Yeah.
This hierarchical structure,
Right.
as resembles to a fractal.
Yes.
Is there a cutoff length below which?
So if you think this as a stiff polymer, right? And again, exactly what's the worm-like chain model equivalent for chromatin? Okay, is an open question. You ask different people, you'll get different answers, maybe in different experiments when they try to fit it different. But broadly speaking, right, the ballpark answer is like a few hundred nanometers, but few hundred nanometers now packs much more DNA, okay, than few hundred nanometers of DNA, okay? So if you take 30 nanometer fiber or something, you most probably can get even 10,000 bases of DNA in the extreme limit, right? So there is some cutoff related to stiffness of the chromatin fiber, right? Under that, you are not going to get it. But above that, really the whole polymer size is the only cutoff, right? This is very different from the molten globule state. In the molten globule state, there is a volume of the polymer, right? And if you track a little polymer segment, it does a random walk and hits the boundary and does a random walk again, okay? So the statistics is not there self-similar all the way. Okay, so yeah. You have to run, Spencer, right?